Now I need to open my
Then you want to
PowerPoint.
share your screen.
Yeah, let me just put it on my, I will put it on here.
No.
And now go back to Zoom
Yeah.
and now share.
See to,
Okay, you want me to get
Okay.
out of here?
I'm just gonna sneak
Sure.
by you for a second.
Thank you guys so much.
And then you should
I
be all
was
set.
so nervous.
You've got it recording. You've got the screen share.
And now the people on the other end, you got to test to see if they can hear you.
She's not talking to them.
You're
She's
not
just
talking to the
using
people?
Zoom to
You're showing
record.
on the other end, like the remote
Yeah,
people.
they should hear me talk.
They may.
Oh, so you are talking.
Yeah,
So if they are not, that's their problem.
Right.
Right?
Okay.
that's right.
Yeah.
And you can communicate through them through text
Ah.
to a chat. No.
So
Yeah.
here, can I close this?
Yeah, you can minimize it.
Okay, so now I just want to see, this doesn't have another, I just have to go. Does this have another field so that I can?
Yeah. It's gonna be in the back. So you'll just have to remember to
Take it off.
Where is
Yeah.
it?
You just had it. That was it. That, yeah.
Yes.
Yeah. I'm just gonna plug it into the back. So just don't remember to take it.
There's two settings on it. Let me see. I think this is power. This is
for the projection or like changing the slide.
change the slide.
Oh. I can't believe it.
Then this microphone, it's off right now. You just gotta hold this.
Yep.
It's on. I can leave it on for
Power
you.
button on the left, the one that says mute.
Okay.
Yep.
Where's the mute?
It's the same button as the power button.
Okay. Okay, so now let's see if this works.
And when it's muted, it'll turn on.
Perfect.
Beautiful.
I'll just put this bar down,
Yeah.
you want, or like on the side so that, yes, perfect. Or on the side somewhere, because you
Perfect.
also have some things there.
Oh, what do
Oh
you
wait,
do now?
nothing. It's just.
Just on the very bottom, if you go back to.
So what do I do now?
go like
Yeah,
this.
There.
All right.
Wait,
Oh, that's not it.
Yeah.
There.
Please, she's got
Yeah,
this.
that's not it.
they do see it. Hide me panel.
There. Perfect. And what's this? Press S curve.
You go off.
Yeah. that-
Okay, guys, thank you.
Yes, of course. If you haven't. Okay.
Please, stop. So I'm excited of meeting in person, not behind the scene. So. I don't know. So I won't very excited.
I thought many of
Who has that option? So, today, I'm practicing to take the exam on its after exam song. And I will tell you story. So last year, guess what the average was? Anyway, 90s, exactly. And it's not because I'm extraordinary. It's because I was doing the exam online. And AI doesn't cooperate with me. I love AI and hopefully did not proceed with signature. So, I cannot say anything. But let's see this year. That's why you have this process. So here are the problems. So, Angela, Suresh, Grace,
Len.
and what is his name? Ross.
Lenny.
Lenny. And Scott. So let's, this is not on that thing. I can see, yeah. And I don't see nations, and what's the thing I can try to ask more. And because today you can have it Talking family, that's pretty good. And it will be more sledding around. Now, if you sits up close. We all like the pilot that sits in the watchbox, exams off. And the advantage of it is one, you don't need Wi-Fi. Actually, today the Wi-Fi is excellent. I used to teach before Providence here every single day for a decade and more. So the Wi-Fi now is vastly improved. But when you're taking your exam, you will have it downloaded on your computer. And then I give you the password, matches, you have access to the exam. So that's what you're going to try to do. I have a mock exam, and it has nothing to do with human physiology. It's just for you test it if you know what steps necessary to take the exam. So how many of you, open everybody, has a hand start active? Can you raise your hands? Plus, so who doesn't? Oh, just a couple. So now I can go to my retro, I can have the steps, you already those. Anyway, can you see the screen well? So these are the steps that you have to take. Now, I don't have because we're all already signed on to exam stuff. Are you all signed on to the market exam? So you can start taking the exam, let's see.
Is it switched on? I don't think it's a PowerPoint. I think it's a screen. Oh wait, it is. Just kidding. It is a PowerPoint.
It is
Okay.
perfect. I mean,
Oh, there you go.
I changed it, but it's not working.
Supposed to.
Yeah.
Is
Before
this switched on?
Maybe you can.
there.
Okay. Oh, there you go. Okay.
So the course, you know, the course, our course is known as Anatomy and Physiology One. Then, when you go on the mock exam, this is the password. So now you all know the password. Hey, there has to be a lot of excitement there. Can you share so we can also have fun? If not, you have to be stuck listening. So this is the password, mock exam 2025. So now it's available to everybody. On September 25, you won't know it until you're all here and I decide that it's fine, we can start the exam, and I will put the password up here. Okay, so now try to access the mock exam and enter the password and start the exam. Let's see what happens. Mock, but people got a case. Okay, it has to be exact. Hey guys, this is supposed to walk around. Unless you like to be here on stage. See if anyone has a problem. Oh, Kevin. See your arm. Oh. So anyone have any problem? You have to enter the password exactly. The two other spaces, the E and the M. Yes. Sorry? No, you have to enter the mock exam. That's it, that you have. No. So you have to be able to access the mock exam. So who has been able to access it again? Okay? Nobody has been able to access the market thing. The second one. Okay, that's the second one, please. Please access the market thing. No? Yes, you can stop. Stop. You can stop. Whoever was starting, don't jump in. We cannot access the example because you don't have what answer. That's something we cannot be clear about. Because how you send an email to everybody. So everybody should have a new instruction. So you can stop the either market there. So if anyone sample computer or your requirements are not compatible, I send you an email, but you can get around from comfort the computer. So that's why my email was very remote online. Comfort was a laptop, well, okay? So that's what I university. They actually university law. And why? Because there's hundred students at the nursing school that are part of our class. And they are going to become this program. So that's why we now have got the Biol nursing school. The ones that are not signing, maybe you didn't go at your email, it was on there. You have a Gmail, and we didn't go at the company email, then it was an instruction. So you have to go back. That's something we cannot make error with now. Furthermore, and some are not compatible with this software. So if you have these smart devices, if you have a Chromebook or Samsung, they're not going to be compatible with Samsung. Go ahead, you have the option of going to the computer, laptop, now, and you can get a computer, which is simply compatible. Okay, so that's the way around. Right, if you have problems downloading the software, that means that you don't have the follow instruction correctly. So remember, if you are not even first. Have your Gmail, that's it, the email, something like that. Or have your quick email. No, they're open. See, we can help some students. You know, gracious. Yes. See, 465,000 students. So we cannot access the program, maybe start looking at my notification. That means you probably could not listen at the right place, because you use a different email. I know you have many emails that understand emails and so on. So you have to use the email that is listed on the first. Okay? You guys see it? So the question is very good, right? When you get on the seat. So, this is a very good show because you don't know. It's new to me, I have to be training, but now I can see, now on, so you learn this. Yes, you can look right here first. No problem. But if it would be the real exam, you put it. So now it's the time to correct the problem. No, you can leave, but I just get started. So, yes, right now, yes, you can go back. But in the new exam, there's not going to be any backtracking. The questions are going to be random and the answers are going to be random. But right now, I don't know if you have to know that class. Then you have to start from the beginning. You can start at certain times as you want. No. Interview exam, no, there's no background. There's no title. You just, it's a multiple choice. You cannot go back. You cannot go back. And the system that I have heard in your computer is simply lifted from the first sentence. Welcome to the development. First, did you get it? Yeah. Well, writing a fiction has no effect. Let's talk about that. And if you want to, read that person to me, because they think that the answer is wrong, you can do that later. So the writing a question is not feasible. Okay, so everyone is done. I can start capturing the more interesting aspect. Does anyone still have any problems in the please raise your hand? You can start talking with me. Actually, there's some problems you can have. Okay, I feel I'm more of a point. No, the flag means something. Okay? You cannot go down. Yes, the question from the beginning, they all multiple choice. No writing. And next week, I'm going to post example of question. And if we have time, I can have a review, an extra review, which already know on Sunday, and I will have class on Sunday. It's recorded. So, What happens? Okay, so everybody saw it? What problems solved? So when you come on screen 25, you cannot have any smart devices with you. So if you have watches, if you have iPhones, they all have to go into that. And you cannot use them because those cannot be God's law. Okay? So you can only use one smart device. You cannot use your phones to use communication there. You can use laptops and iPads. And to repeat, if you have a phone or if you have a something, those are not compatible with the same spot. So you can have to get something outside or you can ask something for God. So if you do problems one maybe we not pushing around the castle and castle. You have to go back, start from the beginning, go to your really first email, because that's where the invitation, my invitation was sent to. Yeah. So, I mean, so you can stop your phone? What is it? Yeah, so. So, I know, I'm moving because you have to sit in the box. And I'm to the plastic. That's what I want. I believe that that's so perfect. Yeah. So what you have to do is access and say, that's it. And what was I saying? Let's just know the difference. It's very cool. So I say second message with new instruction. And those should work very well, as step by step. That's what it's here. These are the new instructions. Did you see this? So you have to go step by step, go back, turn everything off, and start making this here. Now, it's up to them, but I'm going to gather, when you're done, in about 5 minutes, I'm going to start having some lecture. So you can do it off. So. What? Well, this browser that, lot of the last. Right? Okay. So maybe you got these new instructors, step by step. Okay. There's no. I contact the president. I cannot help you with this. Okay? Everybody gets some. I don't know. I don't know. Right. The many of requirements doesn't need to get it out of computer. So, how it takes support, you can have a solution. If you have maybe people know, computer know, computer.
If they're done, maybe.
If you done, it's up to you.
Okay.
I'm gonna discover something here. So it's up to you, you do whatever you want. I have the slides and notes of policy, but I'm not sure if it's really important. Okay? It's up to you. I would like to say, yeah, I know what I'm done, but that's up to you. Right? Yes. If you want to say now, because you have any problem, you can do it not home. You can get your own little over and over again, make sure that everything is properly, also in the file. Okay? So, I would send you, I'm not going to send you. This exam is going to be available 24 hours for the day. If you don't see it, you don't know what it is. You will only see it when I do the class. It's magic. It's okay, so you won't make it process, you won't make it problem. You won't know this. No, no, okay? So the exam itself, patient. I'm going to talk over this. The field class, I can only see long term. You won't be able to do that. Okay? No, no. The other people now are doing something because. Okay, this is going to help out the cities.
You get a lot of sorts. Okay.
So, really help them out. Go to the project, you move it for flows of blue. Okay? They're not to try to help. And if they know that the school, then you come to us in the clients. We're getting things so people because you cannot help anything right. This was the purpose of it. So they can know what we say. So that once everything is working well. I'm going for it in that. Right? I don't know why, you know, excuse me. I know it's gonna be bold, but the next slide, you know, it's very close. Okay? It's good, it's very important, most of kids. So I don't know. So, it's really, the multiple, they are excellent, and finally, the one is something that's. There's a little chat box on the left corner, and they are excellent. Okay? There's no thing. Okay, go on. Right. The new question, I need to tell us self the consent that's not in place. Yes. Yes. I see. No, I agree. After this, when I become satisfied, I'm going to rent it. Okay? So I mean, you guys said nothing. So. So, what is the time? Okay, so I think that we still enough time on the 10th call. So we know the purpose is to know the environment. When we come here, nothing to be sitting near the phone. There's going to be one seat in between. And all the problems should solve. Everything should be resolved. So now, in this September 25, you have lots of time to build tech support. I used it and then have a chat on the Northern Rock Journal. You see that particularly you're going to step by step, you're going to solve it in the part. You're very patient, I listen. So when you more email concerns, the company will tell you the teenager. Because we see the family is not start from the beginning, this is supposed to. We start with instruction, no question. We let him now sit. So though you can use, you can do step by step, because it works for most people. Okay? You know that it's going to be recorded. When the slides, you know, because yes. So how you going to check for the thinking? I would take it from the whole class again. Okay? Because right now, I cannot do that. Okay, go back. Let's start lecturing. Okay. So I'm not back to that. That's stupid. So for the prophets, they feel you can leave, you can leave, but for safety. If you don't have to do anything else, no. Okay?
Have a good day, Maria.
Thank you. Thanks for all.
My pleasure.
Okay, so you start by reviewing where I start, where I ended last Tuesday, when I was talking with Barbara about. So we went to passive pencil. Now we go to the active pencil. If you remember, active pencil requires energy, potential energy. It does not depend on kinetic energy, which is the energy of movement. So active transport requires what you Remember, because it's going up the hill from low concentration to high concentration. And there are two types of active transport. You have the primary active transport and the secondary active transport. So you guys might collect this in person or on Zoom. Let's see who likes in person, raise your hand. And who likes on Zoom? Well, it looks like half and half. So you have the exams in person. But I like Zoom because there's many more questions asked by the students. Okay. So here's the primary active transport. And you see, this is protein, right? These are proteins that are embedded in the plasma membrane. Remember, the plasma membrane is made up, so I need someone to tell me when it's 5 o'clock. Well, let's say 5:05, because I have my watch here. Oh, we still have lots of time. Okay. So, these proteins that are transmembrane replaced, they are very important for active transport, also important for passive transport, like for the transport of glucose. So, here is the primary active transport. So this is protein on its side. It's a pump, but it's an enzyme. So it has two functions. One function is to hydrolyze liquid. Remember hydrolysis is to break apart molecule with water, with the help of water. And here is the secondary active transport. This is also pump, but it's not ATP dependent. So you could say, well, if it's not ATP dependent, why do we say that it's active transport? Why? Because the transport of ions in this pump depend on this sodium potassium ATP. So let me go back. You see here, the yellow are the sodium ions, the green are the potassium ions. And you see that in this pump, fits very nicely two potassium ions and three sodium ions. So the potassium ions go in and the sodium ions go out. And if you remember, the concentration of sodium is much higher on the outside of the extracellular fluid, which would be the interstitial fluid. So the sodium concentration is much higher on the outside, potassium concentration is much higher on the inside. And if you think if you look at this sun, you see that it's potassium ions to the inside. So it's going from low to high concentration, so up the hill, and it's sodium from low to high. So this protein is sodium potassium pump. It has two functions. It transports these very important ions, sodium and potassium. Without this sodium and potassium ion, we would not be able to maintain our disease, because they are very important for the transport of signaling by the neurons. So it transports the ion, but it's also an ATP gas. So it breaks down ATP, the covalent bond from ATP needs energy. So that then the pump can push the ions in against the concentration gradient. Now here, you see that it's a different scenario. Here, so the yellow is sodium, the yellow is sodium, the red is calcium, and you see calcium is coming out, and sodium is going in. But if you notice, the sodium is going from high concentration to low concentration. It doesn't need energy, right? You only need energy to go against the concentration gradient. So what happens is that the calcium takes advantage of the energy generated by sodium to now move gets its concentration there. So I want to make this clear. You see here, the sodium is drawn from outside with this high concentration, low concentration. And this movement of sodium develops energy so that calcium can move from low to high concentration. So secondary active transport takes advantage of the primary transport. Okay, so this sodium potassium pump that I showed to you here is one of the most important proteins in our body. 40% of the ATP that is produced in the mitochondria is used to maintain this sodium potassium pump. So I told you it's very, very important to maintain this differential concentration of potassium and sodium. So outside the cell, there's around 4 millimolar potassium, but inside is 139 millimolar potassium. So the sodium potassium pump is pushing sodium inside to maintain this homeostatic concentration of potassium. Sodium is the opposite. This high concentration on the outside, 145 millimolar. So that pump is pushing sodium outside. You can see here that the sodium potassium pump is pushing potassium outside and push inside. So it's a pump that promotes the movement of these two ions that are positively charged, but they cannot cross the platinum medium because of the polar ends of the phospholipids. So they need to use this pump to get in and out of the cell. So, if you are a nurse in the ward and you see a bottle that has sodium chloride and has potassium chloride, they both have exactly the same color. They don't transfer. But if you put the sodium fluid into an IV, that's fine. If you put the potassium into an IV, you can kill a patient. Why? Because then the concentration of potassium will be very high on the outside. And that is highly toxic. So you might ask, that's so toxic. Why do you have potassium in the hospital? Because some people, some patients, have low concentrations of potassium. So you have to be very, very careful what you put in there. Okay. So active transport, there's different pumps that are used. So you have uniports. Uniports, that means it's only one molecule that's moved. Then you have syncports. That means it's two molecules going the same direction. And I'm going to give you an example. So for example, glucose and sodium, they go through this secondary active transfer. Remember, glucose goes into the cell, facilitated. We talked about these transporters, glucose transporters that are regulated by insulin. But glucose can also be transported by the secondary active transport. And here, so this would be a syncport when the two ions or an ion and a molecule are transported in the same direction. And then you have the antipole, right? The sodium and potassium pump. That would be in the antipole, where sodium goes up, potassium goes in. So you have these three different types of proteins that conduct ions in the opposite direction. Okay, so here again, because this protein is so important, the sodium potassium pump. You see, this is the outside, this is the inside. You see the three sodium going out and the two potassium going in. And you could ask, how come there's two potassium and three sodium? Because the potassium ion has a coat that makes it much larger, and there's only two ions that have fit in this pump. So this is such an important pump. Remember, this is a protein. And 40% of the energy that is produced by the mitochondria is used to maintain this. Okay, so here now we have an animation of what is secondary active transfer. So you see here, the ATP is not directly involved. So here we have this part, it's also an enzyme that hydrolyzes ATP. So it's an enzyme and a carrier. Now here, this transporter of one or symbol, or solid, correct? Where this is a symbol, it doesn't depend directly on ATP. What you see here, you see glucose and you see sodium. And they're moving in the same direction. Glucose and sodium. So glucose takes advantage of the kinetic energy generated by sodium to move inside the cell. So we say that this is this coupled movement of two molecules or ions, which is going in the same direction. And I will explain why this is dependent on ATP, why we call this active transfer. And here we have an antibody where you have calcium, the red, and the sodium ion. And you will see, now we don't see this not working, but you saw before how the sodium goes out and the calcium goes in, right? So, no, sorry, because this is a simple, the high concentration of sodium, the sodium goes in, and the kinetic energy generated by sodium going in allows calcium to go out. But this is the difference. So why is this dependent? Why do we call this active transport? First of all, glucose and calcium are moving from low concentration to high concentration. So that's the definition of active transport. But now, let's look at glucose. So glucose is such an important molecule for making ATP in our body that it has two ways of being transported. So here, you see the symbol where sodium is going in, sodium is going from high to low, so that is not active transport. The glucose is going from low to high. And it takes advantage of the kinetic energy of sodium. But before, in the previous lectures, we said that glucose also can be transported by active transport. So where is this present where glucose is transported by two different mechanisms, one being passive, the other being active? So this is present mostly in the cells, endothelial cells of the small intestine. So let's see why. So this is an endothelial cell. Here you see it's the extracellular fluid. This is the lumen of the small intestine. So the food is coming through there, and the glucose now has to go across these cells and the cellular fluid so that it can get into the blood. So what happens here? What you need to remember is that in this cell, the concentration of glucose is higher compared to the concentration of glucose in the lumen of the small intestine and compared to the interstitial fluid. So you see here this arrow. Here the concentration of glucose is high, corresponds to the intracellular fluid, and at these swells, you have low glucose compared to inside the cell. So now you have glucose in the lumen of the small intestine. And it has to be transported through these cells and end up in the interstitial fluid so that it can go into the blood. So in the lumen, there's low glucose. So it's going from low to high. So here, at this end of the cell, if the glucose goes in using the simple, the active transport. Why? Because it's going from low to high concentration. So it's going against its concentration. Now at this end, you have high glucose going low glucose. So here, glucose is transported, I facilitated diffusion. So this is an amazing mechanism that happens in these cells, so that these two proteins that have completely different mechanisms of transporting glucose. So they are genetically determined. They have to be positioned in this cell, and they transport glucose, one from high to low, that would be the simple, and one from low to high, that would be the facilitated. So this transport of glucose doesn't occur in all cells. Specific to some cells, an example being the endothelial cells of the cell. Okay, so this here shows some examples of these active transport proteins involved. So here you have calcium going from the cytosol to the outside, while sodium goes in. So this is an anti-pore. So this would be an example of a secondary active transport. Here you have sodium going in, hydrogen going out. So sodium gradient goes from high to low. Here you have glucose going with sodium going into the cell with sodium. This is the simple. So this is also a secondary active transport. And now here you have amino acids. They also take advantage of this secondary active transport. Okay, so now I want to describe a very, very important mechanism to maintain the good function of the heart. So the heart needs a lot of calcium. So it depends on this anti-pore. So why calcium? Calcium is very important for muscle contraction, not only for skeletal muscle, but also for cardiac muscle. So the more calcium in the muscle cell, the stronger the muscle cell, the better the muscle cell will function. So it depends on this anti-pore. So you see sodium going in and calcium going out. This is an anti-pore, and the calcium goes out depending on the kinetic energy of the sodium. Because sodium goes from high to low, like calcium is going from low to high. So why do I say that this is a secondary active transport that depends on the sun? This is the sodium potassium pump. So this pump that uses the heat is responsible for maintaining high concentration of sodium on the cytosol and low concentration of potassium base. So if you inhibit this pump, then there will be more calcium inside, because you will have less sodium, so less sodium going in, therefore less calcium going out. So if you think about the cardiac muscle, inhibiting this pump, not totally, partially, would be very advantageous for the cardiac muscle, because you would have less sodium out here, and therefore less sodium going in, less calcium going out, and therefore there will be more calcium in the cardiac muscles, which will help the cardiac muscle. So guess what? There's a product that is developed from this beautiful brain. It's digital or something like this. And what it does, it blocks protein. So it blocks protein with sodium potassium pump. This stuff. Okay, so if I go back to the previous slide, why is it stuck? Oh. Oh, good. There we go. So this plant, the chemical synthesized or purified from this plant will inhibit the sodium potassium pump. Not 100%, of course, because that would be lethal. But it lowers its function, and therefore, by lowering the low concentration of sodium in the output, this will affect this antibody. Therefore, there's less sodium. The sodium will develop less conductivity, and the calcium will stay in the cell, therefore it being used for muscle contraction. Is this clear? It's also very interesting mechanism, and the investigators were able to take advantage to develop medicine for the cardiac muscle. Okay, so now we talked about active transport and secondary active transport that relate to small ions, the transport of small ions. Now, when there are large molecules and microorganisms, they're transported by vesicular transport. And there are two types, endocytosis and exocytosis. So endocytosis, it's easy to remember. Endo comes in. So brings large molecules and the particles inside the cell, and exocytosis, they are secreted. So you can think about hormones that in glands that secrete hormones, they use exocytosis to transport, to secrete the hormones. Okay, so there are three types of endocytosis that bring in things into this. Phagocytosis, this is used by white blood cells to bring into the cell, for example, bacteria. So they're brought into the cell, the white blood cells. That's how the white blood cells work in the immune system. So these bacteria or microorganisms are brought into the white blood cells by endocytosis, and then they are digested by the lysosomes. So you see here what happens. This is a plasma membrane, and the plasma membrane develops these cytopores that will encircle the microorganisms and internalize into the cell, and then diffuse the lysosomes, destroy the microorganisms. So you see here the white blood cell engulfing the bacteria. You see here by endocytosis, you have the phagophore, and the particles are internalized, forming a phagosome that is going to be fused to the lysosome. You see here, this is the bacteria, and the white blood cell circulates. Then you have another type of, let's see if I can move. Another type of endocytosis, which is pinocytosis. Pinocytosis brings things into the cell with more liquids and smaller particles. So it also forms, it involves the liquid and smaller particles and brings them inside. And then you have the most interesting form of endocytosis, that is receptor-mediated endocytosis. Now, this is very, very specific. So this is a way, for example, that viruses get into the cells. So viruses, they hijack our own proteins. They bind to them, they through receptors, and this allows them to come into the cell. So this is receptor-mediated endocytosis. So what you have here is slice-shaped molecules of receptors, and here you could have particles that contain cholesterol bound to these receptors or viruses, bind specifically molecules that are decorating the plasma membrane. And they get into the cell. This is a very highly specific way of bringing ions, particles, viruses into the cell. They have to bind with specific receptor. and they form this vacuum coated pit. They go inside, and hopefully they will also fuse with lysozyme. Okay, so the second mediated endocytosis brings into the cell specifically through their specific receptors, enzymes, hormones such as insulin, low density lipoproteins. These are little vesicles that contain cholesterol. Cholesterol is not soluble in water. So how is it transported throughout the blood? It's transported in these vesicles that contain the cholesterol inside the low density lipoprotein. And they get into the cells via receptor mediated endocytosis. And then you have also virus. So here you see these particles, LDL, the low density lipoproteins. If you do a blood test, one of the things that is measured is the concentration of cholesterol. What does it mean? The LDL, I'm sure you have seen this if you have a blood work. So these particles, they contain proteins on the outside. They also have a phospholipid layer. And inside, that's where the cholesterol is. And they have a protein that binds to a receptor for LDL, that is particle specifically. And when it binds to the receptor, the lipoprotein is internalized, and then cholesterol is released. So it's very important to have low levels of LDL. If this mechanism is not working, then cholesterol stays in the blood and leads to the formation of these plaques leading to atherosclerosis. So these receptors are specific for the internalization of vesicles known as the LDL, which contain the cholesterol, have to be there and working properly to prevent atherosclerosis. Then you have here COVID. So how do this virus? How does it get into our cells and be making so devastating effects? So it gets in, hijacks one of our proteins that is known as ACE2. So it's a receptor that is sticking out of our cells. It's a normal receptor that's used in an enzymatic process. But the virus has a protein that's sticking out of the cell, these are the side protein, and they bind this ACE protein, and then it gets in by receptor-mediated endocytosis. So it's amazing how the viruses develop these proteins that can hijack, bind to our own proteins. The same from HIV. So HIV is virus binds to CP4, and this other protein, it's known as a chemical receptor. And these two have to be together for HIV to get internalized by receptor-mediated endocytosis. So one way to prevent HIV infection is to have a molecule that binds to the chemokine receptor, preventing HIV from getting in. So now let's go to exocytosis. So exocytosis is a secretion, the moving out of different molecules. One of them being hormones and also nutrients and so on. So in this case, you have vesicles that are inside the cell and they move depending on signals. It could be calcium, it could be all kinds of signals that induce the movement of these vesicles to fuse with the plasma membrane, open up, and release it away. Here you see an electron micrograph showing a CP4 granule in a cell that speaks hormone. Here's the hormone granule. So opens up, and the product is released. So this is exocytosis. Another process very important that takes advantage of exocytosis is the release of neurotransmitter. So here you have two neurons. This is the end of one neuron, and this is the beginning of another neuron. So you see that they are separated by what we call synaptic space. So the neurotransmitters, so the interaction between these two cells is not through contact, it's through a chemical interaction. So you see these vesicles that contain the chemical, which we call neurotransmitter, that when there's an increase in calcium, at the end of the axon, the vesicles will fuse and release the neurotransmitter into the synaptic web. Then the neurotransmitter, by diffusion, binds the receptor and continues the transmission of the electrical impulse. So this is also a very interesting mechanism, very important for homeostasis, because the neurons are not in contact with each other. They depend for the transmission of the signal. They depend on the release of chemicals that are stored in the vesicles, and when there's a signal like high increase of calcium, they will move to the plasma membrane and release the content. So one can take advantage of these processes to develop medicine. For example, you know that to treat patients with chemotherapy, it can have many side effects. So it's based on the fact that the cells that are cancerous, they're very, very active. So they're more sensitive to these toxic chemicals than normal cells. So when you treat a patient, for example, with the chemotherapy, one of the effects is they lose hair. Why? Because the hair follicles are highly active. So they are also sensitive to these toxic chemicals. So can one devise a method to deliver that toxic chemical specifically to cancer cells so they don't have an effect on other cells? So what is the strategy here? Is to put a receptor, an antibody, and this is HER2. HER2 is a protein that is present mostly in cancer cells. So here you have a phospholipid vesicle that is synthetically made, and it contains the drug, the toxic drug inside. So then you inject this into the patient, and this vesicle is going to bind only to cells, the cancer cells, that are highly enriched in HER2. So when they bind, they will release the toxic chemical. So this makes this treatment highly, highly specific, not affecting other cells that could have secondary side effects. So another use for this exocytosis is, for example, to make liposomes. So liposomes, again, these type of vesicles, but now they don't have the antibody for HER2. So in this case, they contain melatonin, or they have proper melatonin. Melatonin is a hormone released by the pineal gland, and it plays a very, very important in sleep. So many people that cannot sleep, they take melatonin. So how can we deliver melatonin in a highly specific manner? So again, you make these synthetic vesicles that now contain melatonin, then you inject the patient, and the melatonin is delivered to the specific cells. Okay, so we talked about how the membrane, a plasma membrane, is a very, very useful barrier. How it limits how different molecules get in and out, from ions to bacteria. So it has all these. Oh, yes, question. Sorry? Oh, thank you. So, thank you very much. So, this membrane is essential. So, it limits the cell. It has its phospholipid bilayer. It allows it to be selectively permeable, so that only ions minerals that are lipid soluble can cross through freely, but others that are charged, that are polar, the size depends on size, they are limited, there's limited movement across the plasma membrane. So it's highly selective. And now there's other properties of the plasma membrane. This is the electrical property of the plasma membrane. So remember, I talked to you a lot about the sodium potassium bond. So here you see several of these properties that are sodium potassium bonds, and then also ATP bases. You see here ATP binds to an active site of this bond, and then it's hydrolyzed to ATP, and it releases one positive group that allows for energy. So this bond allows for sodium to go out and potassium to go in. And this maintains high concentration of sodium outside and low concentration of potassium inside. So this differential distribution of sodium and potassium leads to an electrical gradient. So that means it's the outside, because you have high concentrations of sodium, is positively charged. The inside, okay, instead, they have high concentration of potassium, so it should also be positively charged. But in the inside, there's proteins that are negatively charged. So, due to this differential distribution, one side, the outside of the plasma membrane is positively charged, the inside is negatively charged. So the environment, the outside, near the plasma membrane is positively charged due to the sodium, and the inside is negatively charged. And it's the difference in this charge that allows neurons to be activated. Now just think about it. If the charge would be the same, then if a signal, if a stimulus comes along, then there would be no effect because you have the same charge. But what the stimulus does, when that electrical current comes through, the sodium, it opens channels and the sodium rushes in. And what happens, that movement of sodium into the cell changes the electrical current and allows the current to be transmitted from neuron to neuron. So this electrical gradient is established by the sodium potassium bond is essential for the transmission of the electrical signal in the neurons. So also remember that there's the chemical gradient, because we have more sodium on the outside and more potassium on the inside. So the transient brain has an electrochemical gradient. Chemical because of the differential distribution between sodium and potassium, and electrical because of the charge that are generated by the high concentration of sodium on the outside and the potassium. So you see here the nerve impulse being conducted along the axis of this neuron. So this, the fact that the electrical impulse can move along the axis depends on this high concentration of sodium and high concentration of potassium, therefore having a chemical gradient and also an electrical gradient. So if you have too much potassium here, like if you inject someone with high levels of potassium, then that will be lethal. Actually, that's one of the strategies for the electrical chair, or for when a prisoner is lethal, death penalty, right? Death penalty. Sometimes the prisoners are injected with a high concentration of potassium. Because if you have a lot of potassium up here, then you don't have the sequential distribution. Okay, so come to the end. I'll do this next week. The plasma membrane is an amazing structure in the cell that is a barrier and controls the movement of substances in and out of the cell. They have to be depending on the liquid solid rate, the size, the concentration gradient, and the electrical charge. So it's an amazing structure. It is well worth studying it, and it gives rise to the opportunity of developing many drugs that are specific for diseases and so on. So I went here. It's a little bit early, maybe 3 minutes early. Well, I could go one more, a few more slides. So here you have a cell. See the plasma membrane, the box so much now. But now inside, you have all kinds of organelles. And they have a specific functions. And they all work together to maintain the environment inside the cell under homeostatic levels. So you see, for example, the lysosome. These lysosomes also have bilipid, phospholipid bilayer. What do they contain inside? They contain enzymes that digest macromolecules. So remember that I told you that in the stomach, the pH is very acidic. Well, inside the lysosome, the pH is also very acidic, for the same reason, to open up macromolecules so that the enzymes can chew them out into their molecules. Then you have mitochondria. Mitochondria are small organelles. They have two phospholipid bilayers. So let's say they have two membranes. And what is interesting, they also have DNA. And that DNA comes only from the mothers. It does not come from the fathers, because the sperm loses its tail and loses most of the mitochondria. So the mitochondria in the egg are mostly maternal. And based on this, that's what gives rise to the opportunity to do hereditary studies, because you can go from one person to the other, study the DNA, the mitochondrial DNA, knowing that it only comes from the mother. So these are amazing molecules, have all these enzymes that can break down glucose to make ATP. So from one molecule of glucose, you can get 38 ATP from the mitochondria. Then you have here the ribosome. Well, first the ribosome. So what is the purpose of the ribosome? These are organelles, and their function is to make that peptide bond. Remember what a peptide bond is? It's a covalent bond that is made between two amino acids. So when we talk about free amino acids, that means they are free. They don't have a peptide bond linking them. If they have a peptide bond linking them, then we call them residues. So the difference when we talk about residues or amino acids is if they are linked or not by a peptide bond. So that peptide bond is made by the ribosome. So some ribosomes make proteins that are floating around in the cell, in the cytoplasm cell. The other ribosomes are attached to these soup-like structures known as endoplasmic reticulum, or rough endoplasmic reticulum. Why do you say they're rough? Because they have ribosomes attached to them. So they don't have smooth surface. They have these ribosomes attached to them. And what is the purpose of these ribosomes? So these ribosomes that are attached to the ER, or endoplasmic reticulum, those proteins are going to be delivered to the membrane, the endoplasmic membrane. So they are specific to synthesize proteins because one thing that on the membrane, see the plasma membrane or the lysosome membrane and so on. How about the smooth ER? The smooth ER does not have ribosome. So what is its structure? What is its function? Its function is to synthesize lipids, not proteins, because it doesn't have ribosome. So the Golgi complex, this is very important. The Golgi complex you can think about as the mail system in the cell. So it delivers the proteins to their specific location. So that means the proteins are synthesized in the rock here, then move on to the different vesicles here of the Golgi and go to until they have different tags that are attached to them, so that some end up in the plasma, the others end up in the lysosome. So the Golgi is what determines, it's like having zip codes. Proteins are not synthesized by the endoplasmic reticulums that are around. They are the Golgi attached and zip codes signals so that they know where they are going to be delivered. Are they going to go to the plasma membrane? Are they going to go to the lysosome? Are they going to go to the mitochondria? So this all depends on small signals that are attached to this protein. Okay, so I already clear. So what is the difference between the cytoplasm and the cytoplasm? So this is a very important, two very important terms. It should be used properly. So cytoplasm is a fluid that fills in the cell inside the plasma membrane until the nuclear membrane. So the fluid in blue is known as a cytoplasm, where the organelles are floating. The cytoplasm contains all the organelles that are encased in the space from the plasma membrane until the nucleus. Okay, we end up here. I hope you had fun with the exam soft and you will learn. Remember, on the 25th, everything should be perfect. And have a great weekend. Sorry? Yes. So what I would prefer, because I was just trained also. So I think that you're much better off contacting the person. I use it and they're really good. You can use the chat. So you can call them on the phone, or you can use this chat. It's a little symbol that's on the bottom right corner, and they're very good. Yeah. No attendance. No, there's no attendance. It's just so, it's very advantageous to come here and see the environment and make sure that it is working properly. Okay? Yeah. Yes. Because it's the different distribution. So what happens for the signal to go down the axon? Sodium has to come in. So you have these sodium channels all along, and they control the flow of the electrical charge down. Yes. Okay, so what you have to do is go online and look at the site. Actually, I put it on one of your emails that you can contact a company called Laptop Loan, and they will loan you. Okay, is that good? Okay, so as I said, I cannot help you. You have to access the next support group, okay? They will help you, believe me, they are very, very good. Okay? One will be there, you will know where that's you. And then I will be able to answer and voila, you very quick. Okay? Thank you. Okay, so what I suggest. Third grade. I say work with you. Yes, no, work with the humor, see why isn't it compatible with the same software. I think we work with you. Go on the first floor, right here. It's a student outfit. Okay? Because you understand these technical things, I'm not good at. I can send another email, I can send you another invite. If you want.
Yeah, I need the invite.
Okay, so send me an email and I will send you the.
Okay,
Okay, is that good?
I'll do it. Okay, thank you.
Okay. That's it.
So. I know, I know, like. Yeah.
So how do people depend on the receptor? So the receptor, sometimes they're ready for more than one, but they're very specific. And the downstream response depends on the receptor. Sorry? Okay, so what I stress is the well, really, no, I send you email. Did you get my email? So there, under email, tell me how to access flexible, and they are ready to help. So this is so important. But it's important that you look what's saying. What's saying. You don't but it's what's saying. No, it's what's saying. Yeah. But I don't know to sign this. Electronically, so go to them and ask them to send. What is your name? Yeah, I don't know.
But you said that you don't, it's even so long, like, just like.
Yes, I know, but they will contact me. And then I will enter on the exam software. I can enter for each student how much time they are. But I don't want to do this on paper. You go there.
Oh, you
Yes,
okay,
okay,
so,
go to them and then they will send me a message and I will know. Send that.
so you don't, send this one online.
No, you have to go to the office and see what you want.
But usually they want this form.
Yeah, no. I haven't signed it.
Okay. So, that.
Yes. If you have any problems, let me know. Yes. Yes. I did for everybody. I emailed, but if you want, send me an email and I will send it. Oh, but you can send an email before myself. Yeah, yeah. I send it to everybody. How you get to export. Okay. But send an email to me and I will send you the link of the. That's it. Okay. Thank you. I'll go home. Thank you. Thank you. Thank you. I don't know. What's up to you? Are you learning? I don't have anxiety or any normal person. I haven't had it any. You know, you can ask the guy, right? No, are you kidding? I use that a lot. How can I get something saying you're nothing? What's the life? They'll come up with something good and then you can throw your on it. Okay. Oh, okay. Okay.